welcome to the Westin MCAT podcast with your host, Ahmed
and Eric Olson.
All right, welcome back to another episode of the Westin MCAT podcast with Ahmed and Eric. Today we're going to be talking about the eyes and, the surprisingly complicated structure of the eye, given how small it is. And then the ways, importantly, the ways that it can be tested on the MCAT. And I think it's important to understand, from the get-go that, your typical undergrad physiology class is probably going to test the eye in a lot more detail than the MCAT will. Like the eye isn't as heavily tested on the MCAT, but over the course of tonight's episode, we'll sort of run through all the different structures in the eye, what they do, and like I said, how they can be tested. And so I guess we could start from like the outside in, Eric, just walking through, where does light go first?
Yeah. I'll just say, before we jump in, I think, you make a good point about the eye and how we should all be really well prepared for anything coming at us from an eye perspective. Although, in fairness, we could say that about any given concept on the MCAT. We should know everything really well from our classes and things. But I think the eye is kind of an interesting structure in that it fits really well into all the different categories or subsections of the MCAT, right?
Right.
They can ask us about waves and light and photons from a physics perspective, or the electromagnetic spectrum. They can ask us about neurobiology. They can ask us about anatomy from a bio perspective. And then, once we talk about visual processing, which we won't talk quite as much about today, then you can get into kind of a psych-soc perspective as well. So I think, it fits into a lot of different categories. It can come up in a lot of different situations. As getting to your question, when we talk about the eye and we talk about, I agree with you. I think a good way to think about it, we'll talk about structure and kind of thinking about what happens with a photon when you have a photon coming at your eye, and where does it go first? What does it interact with? What's maybe not quite as important from a processing perspective? And so, when we look at the eye from the outside, because that's where a light source is coming at you from, is from away from your eye. And the first kind of most external or superficial part of the eye that interacts with photons is the cornea. And I'll say, Ahmed, that it's interesting the way you pitch. We should really be familiar with the eye because of our classes. I don't think that I really understood the cornea, especially versus the sclera, until I did an anatomy lab and really until medical school. But the cornea is transparent, and that's why it's kind of hard to grasp unless we're looking at a cross section of the eye.
Right.
But the cornea is kind of a lens. It's almost as if it were a contact lens that kind of sits over the front of the eye. It sits in front of your iris and your pupil. And so again, it's transparent. We don't see it. And so it's a little bit harder to conceptualize. But if you can imagine kind of the whites of your eyes are the sclera.
Yeah.
And if you imagine the sclera coming kind of around the front, and where there's no white in the center of the eye, there's a transparent lens that sits over that's confluent with the sclera. And so it kind of touches the sclera, I guess I would say. And that's the first thing that a photon would touch or interact with as far as the anatomy of the eye goes. And I think what's really important about the cornea to recognize, first of all, is, frankly, I would look up a cross-section of the eye. Just if you're not familiar with it, it's just to know what's what.
Yeah.
But a lot of times when we talk about the eye, and we'll get into this, we talk about the lens that sits kind of deeper in the eye. And we think of the lens as kind of this refracting structure, as something that will bend light and help to focus light towards the retina so that we can see clearly. But really, most of the refraction that's taking place in the eye is from the cornea. And it has to do with the difference in the material that is air versus cornea, refractive properties. And it doesn't really matter as much for this discussion. But I think it's 80% of the refraction of light going towards the retina is coming from the cornea.
And
it's not something And it's not something that I think part of why we talk about the lens so much is because the lens can be distorted, and
Right.
it helps us to focus far versus near. Right. So in some instances, the lens will get really thick and circular, and in other instances, it will be pulled thinner, and that helps us
Right.
to focus when we look far away. The cornea doesn't do that, right? It's a fixed amount of refraction that's happening at any given time, and so it's less like maybe it's less pathologically relevant as we talk about like why someone is not seeing well. But as far as like absolute,
Right.
how much is this contributing to refraction? How much is this contributing to our vision? The cornea is really important, is of paramount importance.
Right. And I think it's helpful to understand that there's multiple levels of refraction. And like you mentioned there, how the lens is sort of accommodating, where you're looking, right? And I remember, because I had terrible vision as a kid and still do, but my optometrist, or one of them anyway, was always reminded me of like the 20-20-20 rule, which helps to, if I understood correctly anyway, which is a big if, but basically preserves the function of your lens. And so that rule is basically every 20 minutes, you need to look 20 feet away for 20 seconds.
Okay.
And that sort of helps you refocus on like more distant things, because most people in our day and age are looking like 6 inches in front of them
your face
for a long period of time. So little optometry plug there for all the screen. And that's the other thing too, is it's not just screens, books too. Like if you're an avid reader, same thing applies. Like I remember there's a friend of mine in like middle school, I think, who would always say like, no, like my eyes are gonna be fine because I read. I don't like spend my time on screens. But like it is literally the exact same thing. But anyway, story for another day. But yeah, I think and that sort of gets into nearsightedness, farsightedness, which is actually something that I've seen tested more on the MCAT of late. I don't know if it's just the practice questions I've been doing with students or if it's, a trend to note, but
what kind of, sorry to interrupt, but what kind of questions are you seeing? How are they testing it? What do we need to know about nearsightedness and farsightedness?
Yeah, well, like what kind of lenses, like what kind of contact lenses or glasses would you use to correct these? Sometimes, what does that actually mean? Like, where is the image forming, if not on the retina kind of thing? And so, full transparency, when I was younger, I thought about this completely backwards. I never thought about the issue being internal. I always, for whatever reason, thought the issue was outternal. And it still made sense. So when I thought about nearsightedness, I was like, okay, I'm only seeing things right in front of me. I want to sort of spread that perspective out more distantly. It always made sense that diverging works to fix nearsightedness, and then farsightedness is the opposite. I'm looking way too far out. I want to sort of like bring my focus more proximal. That's not at all how, it's not the actual science behind it, but I guess from a simple just conceptualization standpoint, that works really well. But, I guess internally with respect to the retina, what do those two different things actually mean in terms of like where an image, forms, and what does that mean?
Yeah, it's interesting. I totally agree with like the way you're thinking about it, just in terms of you had like a strategy,
right?
Like a stupid kind of way of thinking about it that made sense to you. And I think that's really the way, not only with this particular concept, but really with anything on the MCAT, especially with things that are difficult for you to think of, like little tricks and little mnemonics to help you remember. What I would think of, and the other thing that I was always getting mixed up was myopia and hyperopia, right?
Right.
And so the way that I think of it, and this, is also kind of like an Eric thing that I did. So I don't know if this is the right way to do it or not. But I would always think of myself, and I wear contacts. I wear glasses if I'm not wearing contacts. And I think, many of us have issues with our vision. Certainly most of us that are younger who have issues seeing are nearsighted. It's much more common, especially in younger people, to be nearsighted. And like you said, that means that you can see things that are close up. You know, you can read a book, you can see a screen fine, but when you look far away, things are blurry, things are distant. And the thing you always hear people say is like, "Oh, I remember when I got my first pair of glasses and I walked outside and I could just see all the leaves on the trees. I could just see every one of them, and it was so incredible." And so that's this idea of like, you know, when you're myopic, you can see close, you can't see far away, things get blurry.
Right.
And the way that I thought of it in my head, and as far as like how the light is bent, where the image is in focus if it's not on the retina. And when we say what we mean that is, you know, if you look to the side of someone's face, they have light coming in like this. And if the retina is here, when the image is blurry, it's either focusing up here or it's focusing back here behind the retina. And so I always thought of myself, and again, that's embarrassing, I don't even want to say it, but I thought of myself as someone who was strong. You know, like I went to the gym, I'm a strong guy, I'm nearsighted.
Not sure where the joke is yet, but keep going.
It's not a joke, it's just I don't want to like share this about myself.
Fair enough.
But what you think of is like, you know, Eric, he's strong, he's nearsighted. When you're nearsighted, you're refracting too much. It's too much refraction. It's too strong.
I see.
Right? And so the light is bent sharply and the image forms in front of the retina.
Right.
Because the light is coming in like this and it's bent so sharply that it doesn't get all the way back to the retina.
Right.
Whereas in hyperopia, your refraction is not quite as strong and the image, instead of forming on the retina here, will form behind the retina. Yeah. And so those are, you know, some things, as you said, can be tested, and it's important to be familiar with that terminology and kind of what's going on there. I would also add, you know, myopia, hyperopia, presbyopia is visual change that occurs with old age. Right. And the way that I would think of that, and we've kind of gotten away from what's happening to a photon as it moves through the visual tract, but I'll say, you know, we go through the cornea, through the pupil, which is the, you know, kind of the black spot in the eye, which is really just a hole, right? And that's where light gets through the outer part of the eye into the inner part. And once it goes through the pupil, it gets through the aqueous humor and into the lens. And the lens is an interesting thing. I kind of touched on it earlier. It sits, you know, behind the pupil and can refract light as well. And it's controlled and changed and, you know, accommodated, as you said, with these muscles called the ciliary bodies. Yeah. And so there are these ciliary bodies that sit outside, kind of around that, kind of not exactly, but kind of around where the iris would be on the inside of the eye, and attached ciliary bodies on the outside, zonular fibers kind of inside of that, and that attaches to the lens. Right. And when those ciliary bodies contract, they change the shape of the lens and change the way that the light is refracted. When they contract, it's kind of counterintuitive. When they contract, I always would think that it would be pulling the lens apart and kind of flattening it out. But that's actually not how it works. When they contract, they squeeze together and they push the lens so that it's thicker and will refract the light more. Right. Now, the reason I'm going through all of this is because I want to talk about presbyopia, which is what happens in old age. And the way that I think of that, again, this may or may not be super accurate, but the way that I think of it is that when you get older, you start to get weaker. You know, you have trouble walking, you have trouble with muscle mass, you get osteoporosis, all of that. And your ciliary bodies are not as powerful as they used to be. Right. So they're not able to contract as well. They're not able to refract that light as strongly as they might have been able to when one was younger, because the lens doesn't accommodate as well as it used to. Right. And because of that, we get presbyopia, which effectively is farsightedness. It's like you can't read very close up. And that's why when you talk to older people, they're, you know, they're always complaining about not being able. That's not fair. They're not always complaining. But sometimes older people will talk about they need to wear reading glasses now. Right. And that's because as we get older, we're not as well able to accommodate, and we can't refract light as well from close up. Right. So we need to use those reading glasses for extra refractive power. Yeah, I mean, it's sometimes difficult to visualize, which is somewhat ironic, but like how the eye works, right? Like the example with the cornea there, or sorry, the ciliary bodies, excuse me, and how the contraction, in a weird way, almost like pushes the cornea as opposed to flattens it, The lens, yeah.
Or sorry, I keep saying cornea.
Yeah.
Anyway, and I think sometimes just like hearing it can be helpful. But now, okay, so the lens has adjusted based on where the object is in front of us. The light now has passed through the hopefully normally functioning lens and has now focused on the retina, which is the next real major landmark from an MCAT standpoint. Obviously, there's the vitreous humor behind the lens, but MCAT doesn't particularly care. So now we're at the retina, and the short question is, now what?
Yeah, it's a good question. The retina has two layers. It has a pigmentary layer that's kind of on the outside of the eye. And again, it's hard without a kind of a cross section here sitting on that we can point to things. But there's a pigmentary layer that helps to absorb some of the light and make sure that it doesn't scatter around the retina and cause us to see stars and double and things like that. Inside of the pigmentary layer, or I guess, on the innermost layer of the eye, is kind of, the neurons and the photoreceptors. And the retina itself has three layers of cells. It has the rods and the cones, which are photoreceptors. They see a photon and they shoot off an action potential that helps us to. It's called signal transduction, right? They get a signal, which is a photon, and they transduce it into an electrical impulse and shoot an action potential down the optic nerve. The second layer is bipolar neurons, which are connected to the rods and cones, and then also connected to the ganglion neurons. And the ganglion neurons have a longer axon. They receive the impulses from the bipolar neurons and then kind of pass them down. And the axon fibers of the ganglion neuron are what form the optic nerve. That's like a big, thick bundle of fibers that leaves the eye and goes back into the brain, that we'll talk about a little bit more in a minute. But Ahmed, can you tell us a little bit about rods and cones and kind of what the difference is and why we might need to know about those?
Yeah, and I think just building off your point there, Eric, one of the things that I always found amusing when I was learning this in my undergrad is we almost process light backwards. Like it starts at the back, moves forward, then into the brain.
It's all a little bit counterintuitive.
Yeah, the eye is just real wonky. And for those that don't know what I'm referring to, so those three layers Eric referred to, the rods and cones are actually at the very back. Then they move forward into the bipolar neurons, then forward yet again into the ganglion neurons, or the ganglion cells, whose axons, like Eric mentioned, become the optic tract, and the optic tract becomes the optic nerve once it enters the brain. Which I always, I even remember I had an exam question, I think in my sophomore or junior year. It was a written answer question, and it was explain what it means when we say like visual information is processed backwards. And it's just, I was so prepared for that question. And anyway,
you were prepared.
Because it was.
It seems like such a vague question.
Well, that professor was fairly good at, like, hey, this is what this means, just in case you ever needed to know it, kind of thing. I was like,
then you know it's coming up.
I see what you're doing.
Yeah.
But no, and for me, in a weird way, like I've always better remembered things that are confusing to me because I just naturally tend to pay more attention to them.
100%.
It's the things where I'm like, I think I know it really well that ends up like kind of hurting me, but anyway, that's another story for another day. But with the rods and cones specifically, like you mentioned, Eric, those are the photoreceptors. And if we break that down, photo meaning light, receptor is fairly straightforward, right? So we're receiving light-based information. And so obviously there's two different photoreceptors because they do two different things. It's important to note that the rods are significantly more abundant. I think if I remember correctly, 80% of the photoreceptors are rods.
80/20 is always a good rule to go by, but I don't know if that's that's the
Yeah,
case here. It could be.
not MCAT testable, fortunately, but basically what the rods are doing is they're responding to low light. So they're really helpful in low light conditions, really seeing, like the different shades of gray, white, black, but not responding to color in the traditional sense that we think of it. Um, that's the responsibility of the cones. And so there's three basic types of cones. Cones that respond to red, which is on the longer wavelength end of the visible spectrum. So at about 700 nanometers. There's green cones that respond to sort of the middle of the spectrum, and then blue cones that respond lower towards like the 400 nanometer range. And so basically, your brain is going to process all of these inputs and effectively use that to create an image. And we'll sort of follow that signal back to the brain and how different visual fields are constructed. But one of the, and I'm curious to get your take on this, Eric, one of the most random debates I ever had at football practice was, what are the three basic colors? What are the three primary colors? And I've always thought about this from a scientific perspective, both with respect to the cones in the eye and also like any sort of hexadecimal system is RGB, right?
Yeah.
And so I've always maintained that the three primary colors are red, green, and blue. And a select handful of people agree with me, but the vast majority of people, I think yellow is supposed to be a primary color, if I remember correctly.
Yeah.
Red, yellow, blue.
Yeah. Yeah.
Do you have any, like, where does that disconnect come from?
Yeah. It's a good question. I think most of us learn the primary colors like as kids in art class.
I must have fallen asleep during that lesson.
You must have fallen asleep that day. I feel like we learned it every year because it's so important to know. But I think like there's a difference between, and I'm trying to remember. I wish we had talked about this before because there's a term for it. But like there's a difference between colors that are like pigments, like paints, and colors that are light, right?
Oh, interesting.
Because for example, like if you mix red and blue, you get purple, right? If you mix, so the idea behind the paint primary color is red, blue, and yellow.
Yeah,
You got me like thinking about it hard. With the primary colors red, blue, and yellow, the idea is like these are more pure colors, and when you mix them together with each other, then they'll form like secondary colors. They'll form other colors.
Right.
So red and blue makes purple, blue and yellow makes green, yellow and red makes orange, right? And those are like secondary colors.
Yeah.
If you mix all those colors together, you're going to get like brown, right? Or dark,
paint,
gross color.
Yeah.
The alternative is like when you think about colors of light, right?
Yeah.
If you mix together red, blue, and green light, it's going to be white.
Right.
Right. And so it's like they're different. They're like different systems, right? Like there's a difference between mixing paints and mixing wavelengths of light. And so I think you're right. Like red, blue, and green are primary light colors. And I want to say like, I'm trying to think, isn't there like yellow and cyan and magenta are like primary colors in printing or something like that? I don't know. But I think it's because there are like different properties of those substances, like photons versus pigments. And so for that reason, like they have different properties, they mix in different ways. But the idea behind it being like you can use these colors to make every other color.
Correct.
And so when you're talking about light, you can use red, green, and blue. Like everything that we see is kind of shades and intensities of red, green, and blue, right?
Exactly.
Because that's what our cones are seeing. Or that's what they're receiving is various intensities of red, blue, and green. Whereas when we're talking about paint, like you can make any color from a pigment perspective with red, blue, and yellow, just depending on, you know, what proportions and what mixtures you use. So I think both are right from our perspective as physicians, right? Like I don't care quite as much about paint, but that doesn't mean it's not important, I guess, right?
Yeah, it's important to someone somewhere, but That was one of the other things I never could wrap my head around is how does mixing everything make white? That makes black in my mind. But in any case, okay, so our cones and our rods have processed this information. Now it's moving along the optic tract into what's going to become the optic nerve. But one area where I find a lot of students get confused is like where information from different places go, right? And what's interesting is that the left visual field is processed in the right eye, and the right visual field is processed in the left part of the brain. And so, can we walk through like that wiring for students? Like, where is everything going? How does this make sense?
Yes. It's another thing that I think an image would be helpful. And so I would certainly encourage everyone to Google like optic field tracts or something like that would probably yield an appropriate visualization. But really the way that you have to think about it is to divide the eyes, not into two eyes, but into four segments, right?
Yes.
And so you kind of split each eye down the middle. And the way that I thought about it, what we have to just touch on before we really get into how it's processed and how it moves or kind of is transported through the nerves is we have to understand that each eye has a nerve, right? The optic nerve, as you said, cranial nerve 2. And it exits through the back of the eye. And then right next to the pituitary gland by the sella turcica, they cross over. And so there's, you have these nerves and then they kind of cross and share some fibers and make an X. Right. And then they come back out and go towards these different tracts of the brain. And so at that X, some of the fibers and some of the signals that we're getting from the retina are being switched across. And so that's kind of my preface for what is going where as far as what parts of the retina are moving to the left brain versus the right brain. The way that I like to think about it, honestly, is to think about the extremes first, to think about the outsides. Yeah. So I think about, the outside of the left eye. And you have to recognize like light is coming at you in a straight line. And so everything that you're seeing that's hitting the left side, the outside of your retina is coming from across your body, right? Right. Everything that's hitting the outside of my left eye is coming from the right side of my body because the light is coming across, it's entering the pupil, and it's going to hit the back left side of my eye. What I like to think of is that everything that's on the outside, the left side, or the far right side is going to go straight back. It's going to go straight back. So everything that's coming from the right visual field into my left eye is going to go straight back and into the left brain. Right. Does that make sense? Yeah. And for me, it makes perfect sense because I'm visualizing that visual in our head. But like you said, Eric, for the listeners and viewers, like definitely look up the picture of this. But you're right. Like it's almost, it's going into the outside of the eye and then straight back, in contrast to if it's hitting the inside of the eye because it's coming from, something on that side. Like if it's the right eye on the right side, etc. Yeah, exactly. So you have to then be thinking about these inner fields. So I'll continue with the left eye. I think let's forget about the right eye for now. And I think if anyone's watching this, it probably looks backwards because I'm using the front-facing camera. But this is my left side over here. So we've talked a little bit about the outside of the left eye. We're seeing things from the right side of the body. It comes in, hits the retina, and goes straight back to the left side of the brain. Let's talk about the inside of the left eye. So if I'm looking straight ahead, things that are hitting the inside of the retina, this medial side, is light that's coming from the left side of my body. Right. It's light that's coming from the left, it's coming through the pupil, it's hitting the medial side of my left retina. And everything that's hitting the medial side of my retina is going to cross to the other side of the body. Right. So everything that's on the medial side of my retina, it's light from the left side of my body. It's going to come in, it's going to cross over to the right side and the right occipital lobe ultimately. And the same thing can be said for the right eye, okay? So in the right lateral, retina, it's light that's coming from the left side of my body. It's going to go straight back to the right occipital lobe. For everything that's on the medial side of my right eye, it's light that's coming from the right side of my body. It's going to cross over at that optic chiasm, and it's going to be processed in the left occipital lobe. I will just say, I wanted to share a story. I had a student I was teaching about this a couple of years ago, and I think the visualization is really important. And it's funny to talk about visualization because we're talking about vision. But I think seeing this, and I think in particular drawing this pathway out, is extremely helpful.
Yeah.
For this particular process, because it can be confusing and it can come up. It can be asked about. It's not a throwaway topic.
Right.
The way that I taught my student a couple of years ago, I was teaching about the optic chiasm, and like I said, the optic chiasm is where we form this X with the optic nerves that they cross over. They share some fibers, and then they come out on the other side and continue back to the occipital lobe. And it sits there next to the pituitary gland. And so in some cases, it has been shown that in individuals with pituitary tumors, that there can be some pressing on that optic chiasm, and some visual field defects because of malfunction of some of those nerve fibers. And if you think about it, what nerve fibers are crossing over? It's the ones from the medial sides of the eye. And so this left medial eye is seeing everything from the far left side of my body. This right medial eye is seeing everything from the far right side of my body. And so it's the peripheral vision, right, that's affected here. And so if you had a pituitary tumor that was pressing really hard on your optic chiasm, then you could potentially have, tunnel vision, where you couldn't see anything on the lateral parts of your vision.
Right.
And so I give this whole example to say that I told my student, some people think that's what happened with David and Goliath, is that Goliath, couldn't see because he was such a big guy. He had a pituitary tumor. And my student said, "What is that? What is David and Goliath?" But I was like, "You don't know about David and Goliath?" Like, I thought that was like, it's a biblical story, but I thought it was like enough in popular culture that people would know. So can I just tell the story really quick, if that's okay, so that people know? So David and Goliath, like, it's the story from the Bible. It's about, these two countries were at war. And the one country, the Philistines, or one people, I guess, they had this like giant guy on their side, and he was, I don't know, it says in the book like how big he was, some number of cubits. He was like 9 feet tall or something like that, and super large and scary. And so he would come out and say, like, pick any one of you other people and like come and fight me, and we'll fight for our people and for our kingdoms, and whoever wins, then like that's it, and that's the winner. And everyone was scared from the other, the Israelites, like the other people, because he was so large and so imposing. And finally, this young boy, David, decided that he was gonna fight Goliath, and instead of like taking a sword and a shield and all this armor, he just took a stone, like some stones in his sling, and he ended up like throwing a stone right at Goliath's head and like hitting him right in the forehead, and he died. And so David won. And the reason it's a popular story is because, like, a David and Goliath story, like Goliath is a big imposing giant, like a corporation, for example, and David is like, some pedestrian or like environmental activist that sues the corporation. It's like big, powerful thing that gets toppled by some smaller entity. And so it's like, it's a popular story. I think people are familiar with it largely, but there have been some hypotheses from some historians that it's possible that Goliath could have had a pituitary tumor, right? Because we know from our knowledge of the pituitary gland that one of the things that secretes is growth hormone. And so he could have gotten really big from this pituitary tumor, and then it caused him to have tunnel vision because it was pressing on his optic chiasm. I think it's all kind of conjecture, and it's kind of fun to think about. I don't know if there's really any evidence of this, but the idea being that, he couldn't see, like he had difficulty seeing because this tumor was pressing on his optic chiasm. And so David was able to kind of like outsmart him because his vision was poor. So it's kind of a funny story.
Yeah,
but a good way to remember the optic chiasm, right? Is that
Yeah, I mean, that's a story I'm never gonna forget now. That's wild, actually. I've never heard that, nor have I ever pieced that together, but that's incredible. And I guess, like, to wrap up, yeah, okay, so the visual information has passed the optic chiasm now and is heading back to the back of the brain, the occipital lobe, the visual cortex, which is also known as the striate cortex because it's striated under a microscope. Um, and so now, how does that structure of the brain take all of this information and give us the smooth output of just sight?
It's a good question, Ahmed. I would love to hear your thoughts on it. I will just say the other thing I think is important to recognize is between the optic chiasm and the occipital lobe is the lateral geniculate nucleus, which is a bundle of nerves in the thalamus. The thalamus being kind of like the relay station for a lot of the sensory information that we're getting. And so it's kind of a stop in the thalamus, lateral geniculate nucleus, excuse me, on the way to the occipital lobe. But Ahmed, tell us a little bit about occipital lobe processing and things.
Yeah. So I think one of the yet another confusing thing for me to understand when I was learning is you don't really see with your eyes. You see with your brain, which is kind of weird to think about. Like, your eyes are just like gathering, it's almost like a vacuum of light-based information. And then, I guess, to complete the metaphor, somebody empties the vacuum and does something with it, right? That somebody being your brain.
Yeah.
And so, like we've been talking about, the information, right, brought in by the left eye ends up at the right visual cortex, the right occipital lobe. So the back right of your brain, right side information goes to the back left of your brain. And essentially what's happening is the neurons in the occipital lobe are processing this information, and then your brain perceives that as sight. So each rod, each cone, based on where that rod and cone was as well, there's both the spatial information as well as the intensity of firing. So a photoreceptor that's more, we'll say, vibrantly excited or stimulated will fire more frequent action potentials, and that the brain will process all these collections of stimuli, which photoreceptor was stimulated, how intensely, how frequently, and then basically use that combination of information to produce an image, right? And so, for example, looking at my laptop, right, if I'm looking straight in front of me, I see lots of colors. I see blue, I see black from the desktop, right? And so if I focus just on those two colors, right, I know the blue that I'm seeing out of my right eye is stimulating, blue cones directly at the back of my eye, and that's probably going to the left brain, and that's going to be, there's likely lots of action potentials in sequence there because it's a consistent stimulus, and my brain is processing that and producing that color in that space. And now, if I look this way, I can still see the blue, but now it's striking a different part of my retina. It's getting processed differently. And so now, based on the receptors that it's striking, that color of blue appears to my right as opposed to directly in front. And so really, that occipital lobe is just sort of the manager of all of these inputs, and it outputs something coherent, which I think is, at a fundamental level, super remarkable, right? Like it is
incredible. It's interesting. I'm thinking about like, and you'll learn this, everyone will learn this when we go through medical school, but you learn the neurologic exam which if you ask a neurologist is super helpful. And if you ask everyone else, we would say it's super complicated and difficult to remember the steps. But part of the neurologic exam is examining the cranial nerves. And so,
Yeah.
we know what each of the cranial nerves does, and there are specific tests that you do for each of those nerves. And the reason I bring it up is because when you're testing the optic nerve, cranial nerve 2, part of what you have to do is visual field testing. And so exactly kind of like we're talking about, there's like a medial side of the left eye and a medial side of the right eye. When you test the visual fields, you have someone cover one eye.
Right.
And then you kind of flash your finger, or you flash 2 fingers, or you flash 5 fingers and say, how many fingers am I holding up? You have them look straight ahead, and then you look at these various parts of the visual field.
Right.
And the reason you have to have them cover one eye is like you say, the brain is such an incredible computational machine.
Yes.
At, taking all these data points and putting them together into this one cohesive image. If you don't have them cover one eye, then they can completely compensate for any loss of visual field on the left eye. It's almost impossible to tell if they can also use their right eye, because the brain can't tell, oh, my left eye is missing vision in this one spot. It will just fill in with information from your right eye. And so you have to take them, isolated, so you can't trick your brain, right? And so it is interesting. Like, you have to use these techniques to say the brain is too smart. Like, we have to control for the brain, because it's going to find a way to outsmart us,
And that child, like little me, was just lots of random experiments. Like I remember I would close my eye, like one eye, and hold a finger in front of me, and like I couldn't see past it. Right. But as soon as I open the other eye, I can see like straight through my finger as if it's not there. Like if I focus on my finger, I can see my finger, but if I look away from it, now it's like it's just suddenly not there.
You see right past it.
And
that's filling in right from the other eye.
Exactly. Like the brain's wild, man. And like, not to get too off topic, but like that's one of the reasons I don't think like, AI or machinery can ever fully replace human intelligence. I think there's just some things that, will never be, we'll say, developed or recreated. Not to say that, AI and things like that aren't immensely helpful. And, it's funny. I was listening to a presentation from a med school dean today, and he was like very pro. Like, he even name dropped ChatGPT. I was like, ChatGPT is great. I was like, wait, really? And like, it's so, for me, it was so peculiar, like, not peculiar, but unique to hear that from a med school dean, because, every other dean that I've ever not had to come in contact with is always, super, weary of ChatGPT. But anyway, the point being, like, I marvel at some of the abilities of the human brain, but that also sort of explains how we can get these wild, we'll say, conditions or wild phenomena that result from just bizarre disturbances to the brain. As a friend of mine and I were chatting at sort of a team dinner the other day, and one of the things that came up was like how, I believe it was this British lady had a fall or something like that, and woke up with an entirely different accent. Still spoke English, but different accent. And I can't remember what the name of the phenomenon is called, but like it's super rare. But just like things like that you almost laugh at, because they're like, there's just no way. But the mystery of the human brain is just,
I mean, even like, yeah, we're a little bit off, but even the like things that we know some about, like I'm thinking about this patient I had who had, we didn't really know what was going on with him. We thought he had some sort of vasculitis, but he had hemineglect.
okay.
Which is like, we know if there's damage to the left brain, then we think about, word processing difficulties.
Right.
Or I guess the dominant hemisphere, I should say. If there's damage to the non-dominant hemisphere, then we think about hemineglect. And it's like, yeah, this happens. But like, I saw it in person, and it is crazy, like,
yeah,
the person like is looking over to their, if they're neglecting their left side, like they're looking over to their right the whole time, and You could
Yeah.
come over and like tap them on the left shoulder and they can't look over there, like they
Yeah.
can't lift their left arm.
Yeah.
And it's not anything that's like wrong with them per se. It's just the brain acting weird. And,
Yeah.
even the things that we like moderately understand to me are very complex. I guess that's why I'm not a neurologist,
Yeah.
If I understood it or, was pretended to understand it, then that's what I'd be doing.
Yeah.
That came out wrong. That sounded like I was saying the neurologists are pretending. In Portuguese, pretend means like had a goal to. Like if I had a goal to understand it, I would be a neurologist. So I just want to clear that up. But anyway, it's just there are a lot of processes that are difficult to understand. I think there's a lot that we have to learn still about how the brain processes information and how it put things together.
Yeah. And I think just that perspective there, that we know a lot, but like we really don't know we definitely don't know everything. I remember a quote, it might have been like from Einstein or Planck or somebody talking about quantum physics, saying like anybody who claims to know quantum physics doesn't know quantum physics. And I feel like the brain is like the exact same way.
One of those things. Yeah.
But in any case, so to wrap up, we sort of worked through now the photon, all the different structures it passes through, and ultimately ending up at the brain. From an MCAT specific standpoint, what are the most important pieces there that somebody needs to know? I think we touched on the wavelengths. Obviously, the different colors are important, what wavelengths they are, and the physics associated with it. What are the more, I guess, biological and psychological aspects of the eye that we should know on test day?
Yeah, I mean, as we kind of wrap up, I guess first of all, I would say that I think everyone without exception should find an actual piece of paper and get some multiple colors and draw out the things that we just talked about. I think the anatomy is important. I would draw the eye, and I would certainly draw the visual fields. That's, I think, where the different colors are going to come in, is to draw those different tracts and where they cross over and where they go.
Right.
I think probably, in my experience, that's where that's where your money is going to be. I would be familiar with it all, but like, retina to optic nerve to optic chiasm to visual processing, like those are the things that I think you can probably bet on getting a question about in some way, shape, or form.
Right.
So kind of the latter half of what we were talking about, I think is probably a higher yield from an MCAT perspective. But as we kind of discussed in the beginning, there are so many different ways, like even just from a photon, right? Like you could have a whole passage in biochem about like signal transduction in the eye, and then you could get a question about like a photon with some wavelength of light, and like what is the frequency of that photon, you know what I mean? Like just things like that. Like you have to be thinking about these connections and how this particular, how the visual system might play into other questions about other topics and things like that. I think it is an area that is ripe for connections and kind of reaching out to other areas to ask questions.
And that's an underlying theme across the MCAT. And I preach it all the time. It's just like make connections, understand how things relate to one another. And like the best crystal example is like amino acids. Like if you understand amino acids and all the relevant connections, you've just understood at least 25 to 33% of
absolutely,
the MCAT sciences. Right. And it sounds simple in principle, but like for those that are listening with, test dates on the horizon, I really urge you to ask yourselves interrogative questions. Like, how does this relate to this? Don't just know what gets you through undergrad, right? How and why is what really separates, truly elite scorers on this exam. And so like Eric mentioned there with the eye, I challenge you guys to make those connections, see how the physics applies, both from photons and from optics, see how the biology applies from action potentials to the regions of the brain, and find more connections maybe that we didn't have the chance to touch on tonight. Any last words, Eric? That sounded
No,
intimidating.
no.
Any closing remarks?
Yikes. No. I mean, best of luck to everyone who's getting ready for a test date. And I think this stuff is important, but I think we've covered it all.
Yeah, and I agree. And on that note, guys, like Eric said, all the best to everybody testing in the near future. Thanks for tuning in, and we'll see you on another episode of Jack Westin MCAT Podcast.